In the first part of our video on DNA as the molecule of heredity, we're going to address the first two of these questions. I want you to understand what experiments, what is the evidence behind our understanding of DNA as the hereditary material. Next, we'll look at the structure of DNA. Of course, it was Watson and Crick who became famous and won the Nobel Prize for elucidating the structure of DNA. We'll talk a little bit about how they discovered that. And we'll discuss what the structure of DNA implies or suggests about how DNA works. And in the second video, we will address the third question, how cells actually replicate DNA. The story of DNA as the hereditary material really begins way back in 1928 with a microbiologist named Frederick Griffiths. He is famous for discovering a phenomenon called bacterial transformation. He was working with strains of Streptococcus pneumoniae that are virulent in mice. So there were different strains in his laboratory. One strain formed small, rough colonies, shown here, and they form small, rough colonies because they cannot synthesize a polysaccharide capsule. And when they are injected into mice, the mice mount a successful immune response against these rough strains, and the mice remain healthy. The second strain he had in the lab was the smooth strain. They formed large, smooth-looking colonies on peachtree plates because they are able to synthesize a polysaccharide capsule. This polysaccharide capsule confers them some protection against the mouse's immune system. And when these smooth strain bacteria are injected into mice, the mice get very sick and they die. So he was doing some experiments where he had the smooth strain cells and he heated them at 60 or 80 degrees Celsius for several minutes. And so they were all killed by the heat. And the proof is that when he injected into mice, the mice lived, because there were no living cells and they had all been killed. There's big surprise came when he mixed the heat-killed smooth strain cells with the rough strain cells. So if you recall, neither the heat-killed smooth strain cells nor the rough strain cells by themselves have any effect on the mice, or at least do not make them kill the mice. However, when in this mixture, when he injected the mixture into the mice, what happened was that the mouse died. Many of the mice died. And when he did an autopsy on these mice, what he found were living, smooth strain cells. So one trivial explanation would be that the heat killing hadn't killed all of them, but that was not the case. He repeated these experiments very carefully, using higher temperatures and longer times to make sure that the heat had killed all of the smooth strain cells. So in every case, if he just injects only the heat-killed smooth strain cells, the mice lived. In every case, if he just injected the rough strain cells, the mice lived. But when he injected the mixture, very frequently the mice died. So he came to the conclusion that somehow in this mixing process, what had happened is that we had a conversion of rough strain cells to smooth strain cells. And this is what he called transformation. That somehow the rough strain cells, the living rough strain cells, had picked up, acquired genetic information from the heat-killed smooth strain cells to turn the rough strain cells into smooth strain cells. So there was a phenotypic and genetic transformation of one type of cell to another in his system. His experiments were quickly replicated in other labs, and a trio of scientists, Avery, McLeod, and McCarty, pursued the question of what is the material from the smooth cells that is able to transform the R cells. And over a period of a couple of decades, they methodically investigated this question, and ultimately they came to the conclusion that it was DNA that transformed the bacteria, because they went through cell fractionations. They found that the polysaccharides didn't work. They found that proteins didn't work, but if they purified DNA as best as they could, it was the purified DNA mixed with the rough cells. So the purified DNA from the smooth cells mixed with the rough cells would cause rough cells to turn into smooth cells. But it didn't work with proteins from smooth cells, and it didn't work with polysaccharides. And to further bolster their conclusion, they treated their transformation preparation with DNAs. Okay, so they had purified DNA, but it wasn't 100% pure, but based on the techniques they had available at the time. But if they treated their active transforming DNA preparation with an enzyme that destroys DNA, then they abolished the transformation activity. If they treated that same preparation with RNase or proteases to get rid of any trace amounts of RNA or protease that might be present in their material, it had no effect on the transforming activity. So based on their evidence, they published a paper saying that it really was DNA that was doing the transformation. Their conclusions were further ultimately verified by experiments by Alfred Hershey and Martha Chase. So this is called the Hershey-Chase experiment. Before I talk about the Hershey-Chase experiment, I need to talk about their model organism. It's called bacteriophage T2. Bacteriophage are viruses that infect bacteria. For some reason that I really can't explain, viruses that infect bacteria are called phage or bacteriophage. Now T2 is a member of a class of bacteriophages that have this type of structure. The beauty of this system is that there are only two types of macromolecules in bacteriophage. They have a protein coat, so all the parts in this diagram outlined in black are made of proteins, and then inside this head is a molecule of DNA. So DNA is packaged inside a protein head, and then we have all of this tail fiber, which consists of protein. So there's only two macromolecules in bacteriophage, DNA and protein. Bacteriophage life cycle, they make their living by infecting E. coli or other bacterial cells. And after this infection, in as little as 20 minutes, what we see is that the cell lyses. So we get lysis of the cell bursts open, and we get progeny phage. So presumably what's happening is that in this period we get the phage genetic material has to be getting inside the cell, and the genetic material, phage genetic material, is directing the synthesis of more phage particles. So the question is, what is this genetic material? What part of the phage is actually getting inside the cell, acting as the genetic material to direct the synthesis of more phage particles? So Alfred Hershey and Martha Chase devised this really elegant experiment. They grew up these bacteriophage in either sulfur 35, sulfur 35, that's a radioactive isotope of sulfur, or with phosphorus 32, which is a radioactive isotope of phosphorus. So if you should recall that sulfur 35 is going to label proteins but not DNA, and phosphate is going to label DNA but not proteins. So we had bacteriophage either labeled with radioactive protein, where all their proteins had been made radioactive, or bacteriophage where their DNA had been made radioactive. So they set up parallel experiments where they allowed the bacteriophage to infect. Okay, so they allowed the bacteriophage to infect. So this takes just a few minutes where the phage find their host cells and attach and introduce their genetic material. This was all done in a liquid medium. They would put it into one of these bar blenders and turned on the bar blender. And in this violent agitation, what happens is that the cells actually don't burst because they're so small, but the phage particles that have been attached to the cell shear off. So now we have just cells with the infected cells now are going to have the genetic material inside. And then we're going to collect the cells by centrifugation at low speed. And at this low speed centrifugation, the cells pellet and the phage particles remain suspended in the supernatant. So the pellet will only contain the bacterial cells with the phage genetic material inside. The remaining liquid will contain the phage particles, that the phage material that did not get inside the cells. So the question is, in which experiment do they see radioactive label inside the cells? Was it sulfur 35 or was it the P32 labeled phage? What they found out is that in their experiment, the P32 labeled phage, the cells infected with P32 labeled phage contained P32 inside the cells. When they did the experiment with the sulfur 35 labeled phage, the sulfur 35 remained outside the cells. So based on their data, they concluded then that it was P32 labeled DNA that was getting inside the cell. So it was the DNA from the phage that was getting inside the cell, and therefore it had to be the phage DNA that was acting as the genetic material and directing the synthesis of phage particles. So then the hunt was on, really after Avery, McLeod, and McCarty published their results. Even before the Hershey-Chase experiment results were published, people began to really investigate the structure of DNA. Many people, although not all, became convinced that DNA is the genetic material. But how could DNA, which appears to be a very simple molecule, contain genetic information? And the answer had to be in the structure of the DNA. So to understand how DNA works, they really needed to understand what the structure of DNA was. So a lot of people were investigating the structure of DNA. One of these was a man named Chargaff. He isolated DNA from lots of different organisms. So these are Chargaff's results, or his data on DNA analysis. So for DNA from many different types of organisms, he analyzed what are the percentage of the different bases, A's, G's, C's, and T's. Okay. And here's his table that I'm not going to go into. But what he noticed is that in a really wide variety of organisms, he found that the percentage of A bases was equal to the percentage of T, percentage of G bases was equal to C, and overall the percentage of purines was always equal to the percentage of pyrimidines. Okay. And this is true for all cells. The only exception was this phage, and this has single-stranded DNA. All the cells have double-stranded DNA. So DS is for double-stranded, SS is for single-stranded. That means that all these cells have DNA molecules as a DNA duplex. These phage have DNA molecules only as a single strand, not a duplex. That's getting ahead of myself, but I just wanted to explain why Chargaff's rules did not apply to this phi x 174. So whatever the structure of DNA was, would be, it had to satisfy Chargaff's rules. Rosalind Franklin was an X-ray crystallographer. She started working in London, in university in London, and she was one of the pioneers of this technique called X-ray crystallography. She was able to get highly purified DNA preparations and get X-ray crystallography patterns. So this is a very famous X-ray crystallography pattern that she produced, and Watson and Crick obtained a look at her X-ray crystallography pattern, and without her knowledge, but that's a long story. And based on her X-ray crystallography pattern, they played around with various models and came up with the now famous Watson-Crick model of DNA. So let's talk a little bit about this Watson-Crick model of DNA. What they found is that their model fit all of the data, including Rosalind Franklin's X-ray crystallography data, if they put the phosphates on the outside. So there's two strands of DNA, and these two strands are anti-parallel. Okay. What do we mean by anti-parallel? Well, each strand of DNA has a direction, and this direction is most easily seen if you look at the sugar phosphate backbone. So here is the sugar phosphate backbone of DNA with the bases in the middle. So in the Watson-Crick structure, the sugar phosphate backbone are on the outside, the bases are in the middle, and the bases, the two strands of DNA, are held together by hydrogen bonding. So this is all hydrogen bonding. And if you again look at the sugar phosphate backbone, what we see is that one end of the molecule has a phosphate attached to the 5' carbon of ribose, and the other end of the molecule has a 3' hydroxyl end. Okay. So this is what we mean by the directionality. One end of the molecule has a 5' phosphate, and the other end has a 3' hydroxyl. So this is one strand. If you look at what we call complementary strand, so we will draw the direction with an arrow, 5' to 3'. If we look at its complementary strand, or its partner strand, what we see is that the direction of this strand is exactly opposite. And that's what we mean by anti-parallel. They're in opposite directions, because the 5' end here is at the bottom, and the 3' end is at the top. So the two strands of DNA in a DNA duplex, Okay, so a duplex means two strands of DNA that are partnered together by hydrogen bonding between adenine and thymine and between guanine and cytosine. Okay. So this satisfies Chargaff's rules because adenine always pairs with thymine, so therefore in any duplex DNA, the percentage of A will always be equal to the percentage of T, and the percentage of G will always be equal to the percentage of C because guanine always pairs with cytosine on its opposite strand. Okay. So these two strands of DNA that can partner together so that A's pair with T and G's pair with C, these are called complementary strands. Okay. So this is sort of a flat two-dimensional representation. Here is what an actual 3D representation looks like as a rotating GIF. And you can see the sugar phosphate backbones on the outside, and the bases that interact by hydrogen bonding are flat and stacked in the middle like the rungs of a ladder.